Happy to see that you could join us here today. I just have a brief introduction to today's webinar. So welcome to everyone to today's Café Scientifique, the public outreach webinar of the EU Horizon project PrecisionTox. My name is Leonie Müller. I'm the PrecisionTox communication coordinator and your host this afternoon. For this edition of the Café Scientifique, we are going to hear a presentation by Berridge, who will take us on a deep dive into the fundamental scientific challenges that we as a community need to address to advance the paradigm shift in chemical safety assessment. His talk will approximately take 30 minutes. This will then be followed by a dedicated Q&A session where everyone can put their question in the Q&A section or in the chat. I will monitor both channels and then make sure that you will get Brian's and also John's perspective on your question, because John will join Brian for the Q&A. If you have any other questions or technical issues, you can also send that through in the chat directly to me. For everyone who joined just now, this meeting is going to be recorded, so everyone who can participate live has a chance to listen to it at a later stage. So just to keep this in mind. And now it is my pleasure to introduce our speaker for today. Dr. Brian Berridge is the principal consultant at B2 Pathology Solutions. Prior to his retirement from the NIH in 2023, he was the scientific director of the Division of Translational Toxicology and associate director of the Interagency National Toxicology Program at the National Institute of Environmental Health Sciences. That was a mouthful. Brian is a veterinarian and board-certified veterinary pathologist. Brian spent 17 years in the pharmaceutical industry as a toxicological pathologist with specialty expertise in cardiovascular pathology and translational animal research. Brian is currently a member of the advisory committee to the Division of Earth and Life Studies at the National Academies and the Stakeholder Advisory Group for the EU Horizon 2020 project PrecisionTox, where I had the pleasure of hearing him speak before. So I'm happy to hand over the virtual microphone to Brian and then hear more about the topic of today. Thanks. I will stop sharing now.
Great. Thank you, Leonie, and good afternoon, everybody. Let me pull up my slides here. Are they in the proper presentation mode, Leonie?
Yes.
Very good. Good. So again, good afternoon, everybody, and thanks for the opportunity to come and consume 30 minutes or so of your life you're never going to get back, but we'll try to make it as entertaining as we possibly can. For me to share some perspectives as Leonie has just introduced you, as somebody who's been a purveyor of the traditional approach for a long time. But I've gotten very, very interested in an alternative approach. And I have to say that through my career, I haven't felt like that my role has been threatened near enough. And so I'm very interested in challenging our current approach and trying to facilitate our ability to get more quicker, more efficient, broader uptake in some of these alternative approaches that we're all very interested in. So the focus of my presentation is going to sort of bring in some perspectives of the way that I have practiced drug safety assessment and chemical hazard assessment over the course of my career, and some things that I think we can learn from that ultimately are things that we need to consider contextually in the way we're doing developing these NAMS-based approaches and maybe represent a bit of a weakness or a gap. So let me sort of talk through that, and I'm looking forward to the conversation that we'll have after this, during which I hope you all will challenge me, because every time I talk about this, then the challenges help me to make this better, and it evolves my thought process. So first off, let me confess to a few things. One is as Leonie just told you, I'm currently an independent consultant. So when you say that I'm a principal consultant, I'm the only consultant in B2 Pathology Solutions. So that's just me. But part of what I do is actually to support folks who are developing novel assay systems. And again, what I try to do is to help folks understand, what kind of biology should be represented in those systems, what kind of endpoints you should measure in them, how it might be positioned, particularly in drug safety assessment, and then ultimately how to represent that to stakeholders, sponsors who would use those systems, and even regulatory folks. That's sort of what I do. I'm not actually representing any of those organizations in this particular presentation. I'm not being compensated for any of that. I'm really here as somebody who's very interested in our success in evolving this new way of working, but also as a stakeholder advisory group member for the Precision Toxicology Project. Also, as Leonie told you, I've worked mostly in pharmaceutical drug safety assessment, but I've also done time, as it were, in chemical hazard assessment, particularly at NIH and the National Toxicology Program. So a lot of when I talk about these kinds of things, I think there are shared principles and concepts, and I bridge across those two areas, but I'll put a particular emphasis on the chemical side of this because I know that's the primary interest of this group. The examples I will use, I will confess, are going to have a bit of a cardiovascular bias because those are the kinds of slides that I have because I teach that stuff and I spend a lot of time talking about cardiovascular related things. But I don't think it's irrelevant. I think they'll illustrate the point, and hopefully you'll see that. Before I do that, I want to give a shout out to Laura Holden, who introduced a concept in a very concise way at our last stakeholder advisory group that was just kind of an epiphany for me. Because as I'm sure many of you do, I think more about the science that we're trying to represent and the biology that we're representing in these assay systems that we're developing. And Laura shared that there are some things that are sort of parascience, as it were, that are very, very influential in the ultimate uptake of a new way of working, whether you're talking about NAMs or any other novel approach. And it was represented by this Venn diagram. So Laura shouldn't be held responsible for the diagram because that's all Brian and his simple way of illustrating things. But it was this notion of trust, understanding, and confidence. And we could spend the rest of the day talking about those three things. I'm not going to do that. Laura and I are actually comparing notes on how to have a bigger discussion about these kinds of things because we just think they're that important. But what I did want to do was to share with you that I feel like that what I'm going to share with you today, at least my perspectives on this, specifically relates to the confidence part of this equation, right? So what's the reason to believe that I would provide to a stakeholder that would allow them to make the decision to start using these novel approaches and ultimately make decisions? And so I wanted to be able to use this initial framework that Laura has come up with that I think is an interesting way for me to show you where I think this fits in the bigger picture. So first off, one of the things that, well, I'm going to tell you this too, that you're probably going to get tired of hearing me say the F word, frameworks, and the Q word, questions, throughout this presentation. But that's sort of the point of the presentation. And so I apologize that I'm going to use those terms liberally. Part of my initiation into thinking a lot about this came probably 10 years ago when I was in pharma and I led the Microphysiological Systems Working Group in the IQ Consortium, which was a cross-pharma working group that partnered with NCATS at NIH to support their tissue chip project. And many of you are probably aware of the significant funding that they put in to stimulate the development of these complex in vitro systems with the idea that if the biology was more complex, then we would believe that it was more in vivo relevant. And I think there's a fair bit of truth to be had to there. But my first introduction to the portfolio of investigators that they had that were developing all these novel systems was I thought they were brilliant solutions to problems that we probably hadn't defined very well, particularly in the context of the questions that we were asking of those systems. And I can tell you that based on our traditional approach of using animal studies, we have had the luxury of not specifically articulating questions. We kind of understand what we want to use the information for as far as drug safety assessment and hazard identification and characterization and risk assessment and all those kinds of things. But the specific questions in the modeling platforms we use, we didn't define. They're sort of defined on the back end by the endpoints that we measure. And we've gotten comfortable with those endpoints, and I'm going to talk a little bit more about them here in a minute. But as a consequence of that, I think we've got a big gap, and that is that we haven't all agreed on the questions that we're asking, and to some degree, the problems that we're trying to solve. Now, I think everybody has a sense in their head about some form of the problem, in that, I don't think we ought to be using animals, or I need a system that's much more higher throughput because of where I need to use it, or I need something that's more predictive of a human outcome. So there are all these kinds of things that are floating around in our head, but we never aligned on, said, look, this is the problem that we're trying to fix. And relatedly, we didn't define the questions that we need to answer that would help us to fix that problem. So the things that I'm going to share with you today are intentionally intended to explore these ideas of how do we define the questions? How do we define the biological scope of the assay systems that we're designing? How do we know what endpoints that we should be measuring in them? How do we relate these novel endpoints to the way that we've traditionally done model toxicity in the traditional animal perspective? And so I could have started with this and been done with this slide, but I think more succinctly, I think we've got the cart in front of the horse, and in a lot of respects, and now I think it's a good time, as we're sort of wringing our hands over the barriers to getting broader uptake. I think we need to think more about what it takes to give our stakeholders a reason to believe. And so that's a lot of what I'm going to talk about today. So from a problems perspective, I think in the chemical space, it's pretty clear. We live in a world with very, very productive chemists. And unfortunately, we're being introduced to exposures in our environment much more quickly than we can actually identify whether we should be concerned about them. And so we've got a volume issue in chemical hazardous systems. So clearly the solution, the problem we're trying to solve is this throughput issue. How do I do hazard identification and characterization in a meaningful way at pace with the way things are being introduced into the environment? And that's in a context of an increasing recognition that a lot of those things are problematic, and not necessarily problematic in causing direct toxicity, but in fact exacerbating pre-existing conditions, or acting in concert with other exposures, co-medications, pre-existing disease, all those kinds of things that are probably not being modeled very well in our traditional normal biology kind of animal studies. And so to me, that emphasizes the need for us to have a much more mechanistic understanding of the bioactivities of these xenobiotics, because then that allows me to extrapolate them to a much more complex context, which is the human condition. And so I think that's a bit of a gap, and that's part of the problem that we're trying to solve. In the drug development side of this, there's a related problem, but for maybe a different reason. And everybody knows that drug development takes a long time, it costs a lot, and you fail most of the time. It's not a very satisfying business, quite honestly. I learned a lot doing it, but it's not incredibly satisfying because there's more failures than there are successes. And as folks have looked at that, one of the places that they point their finger at is the animal studies that we do. Well, develop a lot of evidence that suggests that I've got a safe and effective medicine, and I get in the clinics and I find out that that's not the case. And so unfortunately, you've then experimented in human patients for without any real hope of benefit and the potential for harm. Unfortunately, because of the throughput of those studies aren't happening until late in development. And so your real opportunity to design a molecule that's going to have that balance, the balance of safe and efficacious, is lost by the time you find out that the wheels are falling off, and because you've got real toxicities. And so even in the drug development process, we need a hazard assessment, safety assessment process that has higher throughput, that's more mechanistically informative, that can actually influence the way we design the molecules that we're designing in that space. The other problem we need to figure out how to solve is the fact that the existing system, so we're not starting from, we'd almost be better off if we were starting from a blank slate, but we're not. We're starting from a process. We're trying to reinvent a process that's been in place for decades, for which there's a lot of trust and a lot of confidence, even though it's not perfect. There's a fair bit of confidence that we generate data that's meaningful and we know how to use it, and most of the time we protect people from harm, even when we recognize that, one, that's not always true, and two, it doesn't have the throughput that I need to be able to solve my chemical hazard assessment or even my drug safety assessment problem. But when you look at the kinds of things that we measure in those whole animal systems, and we're generally using more than one species, so you've got two complex mammalian biological substrate for which we're measuring a lot of different changes in tissues, making up all the kinds of words that pathologists use to describe those changes, weighing a number of organs and measuring clinical pathology and hematology kind of endpoints. There's a lot of data being generated in those animal species over time, over doses, in ways that look a lot of like the ways that we characterize disease in the clinics. And so it's easy to translate this kind of data into that clinical setting, whether you're talking about a citizen who's being environmentally exposed or a patient who's being exposed as a course of treatment. If you were going to reinvent this process, you'd have a very difficult time. There's too much represented in this. But I would contend that we don't have to replicate the entire organism or even this full breadth of endpoints, because we've been doing this for, as I said before, for decades. And the reality of it is that where problems happen most of the time is a significantly smaller subset of things that we're actually looking at every time we do an animal study. And I think that's something that we can leverage. The other thing is, and I'm going to apologize to John because I didn't realize John had as much engagement with writing this as I did until our last SAG meeting. But I'm going to respectfully challenge the statement that we've got the science right, and then really the challenges to getting the adoption is non-scientific, because I'm not convinced that we have solved all the scientific problems or filled the scientific gaps, maybe I'll put it that way, in our developing a NAMS-based approach. And so, again, that's part of what I want to challenge you with today. And I think it's illustrated by this paper. So this was a paper that the FDA put out a couple of years ago. And what they did was they wanted to understand how sponsors were starting to use human-induced pluripotential stem cell-derived cardiomyocytes in their regulatory packages. And so they went back and looked in where that kind of data had been submitted and looked at the specific issues that were being characterized or investigated in those, and then ultimately the impact of having generated that data. And it turns out that there were a number of different issues from electrophysiology to cardiomyocyte injury, kinds of things that sponsors were trying to put a bit more human context around by using these cells. And there was a fair bit of variability in the ultimate influence on the regulatory decision making. So, as an example, in places where we had a fair understanding of the biology and the toxicology, things like arrhythmia and in particular QT prolongation, these kinds of assay systems were very influential. In other places, particularly as it related to direct cardiomyocyte cytotoxicity, not very influential at all for reasons that are illustrated here in the red lines. One, not understanding the etiology of the specific cardiotoxicity, and two, not understanding the predictivity of those cardiomyocyte-based systems for the ultimate clinical outcomes. Well, that's problematic. If that's the bar that you have to get over, we virtually never know the specific etiology of most of the toxicities that we describe. And that's not an entirely fair generalization, but I'll talk about that here in just a minute. And as far as the predictivity goes, that's problematic because we intentionally don't take things into patients that we know are going to harm them. Well, that's not as true for things like arrhythmias that are easily monitorable. So we can generate that kind of human-based data and then benchmark against that data. But a lot of the toxicities, particularly those that involve direct cardiomyocyte toxicity, you're not going to take into a patient because the intent is not to do harm, particularly irreversible harm, which is what cardiomyocyte injury would be. So I'm saying that to say that the reality of it is that the current systems can be influential in places that we truly understand the biology and the predictivity of those assay systems, and not so much in those places that we don't. And so that's problematic when you're designing a system for which most of it is based on not understanding the predictivity or not understanding the specific etiology. And as an example of that, I often talk about anthracycline-induced cardiotoxicity. The reality of it is that the most studied xenobiotic-induced cardiotoxicity known to man is anthracycline cardiotoxicity. And we still speculate on specifically what the etiology is. We know what the apical outcome is, which is heart failure. That's the thing that we're worried about. But we don't completely understand how a time-limited, dose-limited exposure for some discrete period of time in the course of treating a cancer patient ultimately leads to heart failure, congestive heart failure, years later. So again, if the bar for us being able to sell a novel approach is a complete understanding of the etiology of the things that we're putting in those modeling systems, then we're in big trouble. And so I'm rationalizing that there are other ways of sort of working around that particular gap. The other part of this is the questions, right? So there are fairly well high level questions that are defined. We know what we're interested in, right? We're interested in identifying putative hazards to people and at what exposures those hazards would be, characterizing those hazards. As hazards, we're talking about adverse bioactivities. So not every bioactivity is adverse. We're interested in the stuff that's a negative health effect. If you look over on the drug development side of this, it's a little bit more nuanced in that we're intentionally taking things that are bioactive into patients with an intent to manage, mitigate, and as much as possible prevent the potential negative effects of those drugs because they have a benefit side to them. So there's a risk-benefit equation that goes with therapeutics that's not necessarily there for environmental chemicals. And so again, in the course of us doing our animal studies, we generate a lot of data that informs these kinds of decisions. What's missing is specifically the kind of data that you need to be able to answer these questions. So I haven't heard anybody propose that we change the fundamental questions that we're asking on the back end. So we need to have a better understanding of the questions that we're asking to generate the data that allows us to make those decisions. And right this minute, all of those things are defined by animal studies, and they're defined by the endpoints that we've chosen to measure in those animal studies. If we're going to reinvent this process, we need to have a better understanding and ultimately be able to relate to our stakeholders how these endpoints that we're generating ultimately are going to answer those questions and allow them to make the same kind of decisions that they've been making. And again, some part of this is what scope of biology do I actually need to model? Which target organs do I really need to measure? I don't think we need to replicate the entire mammalian, the complexity of the entire mammalian system. But I think we can leverage our experience to know where toxicities are more likely to occur than not and focus on those particular things, which is much more tractable. So a lot of what I'm going to talk about is pointing at frameworks that are sort of growing in their definition and to try to convince you that using these frameworks allows us to start building a bridge from these very novel endpoints and modeling systems to ultimately the kinds of data that a stakeholder, a risk assessor needs to make the kinds of decisions that they're trying to make. And so I'm going to talk through these things briefly. The first one is actually the one that instigated Leone's interest in having this webinar. And this was a paper that we published earlier this year where I got together a small group of folks that were much smarter than me. And I introduced them to the premise that, hey, look, I really think that we've got a vacuum here or a gap in having defined the questions, the biological questions that we really need to answer that will allow us to define the right modeling systems and ultimately the right data and endpoints that would support a risk assessor. So that was kind of the basic premise. And one of the things that came from that, and one of the things you're going to see repeatedly as I talk through this is that I'm not going to be presenting anything that's going to be a huge epiphany for anybody, right? All these things are obvious, and that's the point, right? Is that if we just reflected on the kinds of things that we commonly measure and understand how they relate to the things that we're trying to do in a novel way, then we'd have a better chance of building this bridge from, a novel approach to pretty traditional questions. And so in this particular case, through lots of discussions that went round and round, this took over a year worth of discussion for something as simple as this. So that sort of gives you a sense of the complexity of the problem here. We ultimately decided that the fundamental pillars of toxicity were bioavailability, bioactivity, adversity, and susceptibility. And again, no huge epiphany here. it's obvious that if something's not bioavailable, it's not likely to be toxic because it won't be bioactive, and it can't be adverse if it's not bioactive, and the host wouldn't be susceptible to it. Once it gains access to the biological system, is it bioactive? Is the host actually even seeing it and reacting to it? That's fundamental to toxicity, particularly if that response is an adverse response or a harmful response, which is what we're ultimately interested in. And then again, to consider the varying susceptibility of those hosts. So related to that, currently in our hazard assessment approaches for things like pesticides and drugs, we have identified some specific areas of susceptibility, life stage susceptibilities, like reproduction, for example, pediatrics, for example. But there's lots of other susceptibilities that we're not commonly modeling. Things like geriatric or aged susceptibility, or co-therapies, or comorbidities, or genetic susceptibility. Completely not in our current paradigm. So we made a choice that there were some susceptibilities that we were willing to consider and others that we haven't. Some part of that is just the technical tractability of trying to do that. I think we have some capabilities now that allow us to address some of those susceptibilities. But the last thing we want to do is take a big, expensive current approach and make that bigger and more expensive. And so in being able to incorporate these novel susceptibilities in a higher throughput, more mechanistically informative system is an opportunity to actually improve on the current approach, at least in the context of susceptibility. So again, introduce the idea that there's some fundamental pillars. And in fact, part of the intent of this was to recognize that you could use these in sort of a sequential fashion, right? Particularly if you needed to screen a bunch of chemicals. Bioavailability is a pretty easy one to do an initial screen of. And if it's not bioavailable, then there's no point in evaluating its bioactivity, its adversity, or understanding whether you've got a susceptible host. And so each of these sort of represent the opportunity for an off-ramp. If it's bioavailable, probably need to understand if it's bioactive. If it's bioactive, you got to decide whether it's adverse or not, and then ultimately who's susceptible to it. So again, very basic pillars or things that we needed to consider in designing this novel paradigm. This allowed us to start articulating the specific questions that would underpin those things. So what does it mean to be bioavailable? So those specific questions ultimately start defining specific assessment or test or modeling kinds of strategies. So that was the point, was if I understood exactly what it is that I needed to have these pillars of toxicity, I could start to define questions, and then I could start to define the assay systems that I could use to answer those questions. So that's basically the intent of this particular framework. I keep mentioning the fact that I think that the current breadth of biology represented in our modeling systems is intractable from a much more higher throughput, much more reductionist kind of an approach. This represents the fact that when you start looking at where toxicity generally occurs from a target organ perspective, it's actually a definable number of specific target organs where most of the problems occur. I think there's the opportunity to mine our experiences, recognize where those points of toxicological convergence are, at least from a target organ perspective, and focus on those particular organs. That's how you would rationalize why I'm looking at the heart and not the adrenal cortex or some other organ system, because I've modeled toxicity enough to know that it's more commonly going to happen in the heart than 30 other of the tissues that I look at every time I look at an animal study. The next framework was one that came out of the HESI Cardiac Safety Technical Committee, and this is an idea of what we call the failure mode framework. And this I'll give a shout out to Norma Stockbridge. So Norma Stockbridge was my co-chair for the HESI Cardiac Safety Technical Committee for a long time. And Norman was not a huge fan of animal studies because he felt like that we identified problems in animals that weren't really problems in people. And so we were throwing away really good drugs. And so Norman was a big fan of having much more higher throughput mechanistic kinds of approaches that would allow us to have much more of an impact on molecular design. And so the premise for this is that if we focused on building assay or screening systems designed to detect how we know a traditional target organ fails because of our long clinical experience, that would be the places where toxicity would occur. And you could design a higher throughput system and use it earlier in the process. So the basic premise for that is that most of the target organ systems that we're most interested in, we have a pretty good understanding of the biology of those systems, at least at a clinical level. So we know what cells make up the cardiovascular system. We know what functionally how the cardiovascular system works. We know the sort of biochemistry that drives it, the calcium handling that drives contractility, the ion channel activities that drive the rhythmic contraction of the heart, the energetics, and all those kinds of. So we know a lot about the cardiovascular system, and these are the kinds of things that need to be represented in our modeling systems. Well, the other part of that is that we've been defining or identifying, observing toxicity for a very long time. So we know what toxicity looks like, and quite honestly, toxicity looks like any other disease process or insult to that particular organ system. So it's not like there's anything that I describe as a pathologist that is pathognomonic for a drug-induced toxicity. It's just the way that organ system responds to insult. Things like cardiomyocyte injury or hypertrophy, responding to an increase in hemodynamic demand or some arrhythmia, some disruption in the rhythmic contraction, some change in the force of that contraction. All those kinds of things represent the way the cardiovascular system ultimately goes awry when it's been insulted by a chemical, by disease, or anything else. And so by understanding the biology and how that biology fails, we can design systems that allow us to identify those things. Now, the rub in all of this is, and what's missing from our current approaches is that, again, as I mentioned to you earlier, that one way toxicity presents itself is actually exacerbation of a pathological process that's already happening. And that doesn't get modeled in our current system. But if I had a better understanding of the bioactivity of the xenobiotics that we're looking at, chemicals or drugs, and I understand their sort of modes of action, I'd be able to extrapolate into those kind of complex disease states and be able to decide whether I think that's going to be a problem or not. An example of that, you see a drug that increases triglycerides, for example. We know increased triglycerides is a risk factor for coronary artery disease. I don't have to model that. We don't model that, but we know that's a risk, that's a potential hazard, and so we stay away from those kinds of things. Well, these basic fundamental things that we describe all the time in the cardiovascular system are essentially the failure modes. And so these are the kinds of things that you need to look at the breadth of those failure modes to be able to completely cover all the possible ways that toxicity can play out. And in fact, there are examples of toxicants that do all of these things. This is a paper that just came across my desk this morning, and so I quickly made this up because I think it is illustrative. And I'm going to pick on Joe Wu out at Stanford, mostly because I know Joe wouldn't be at this meeting, so I can get away with it. And two, he's a brilliant guy, and so he wouldn't be offended that I'm using this as an example. But they designed a system to screen for novel therapeutics. And in this particular case, they were looking at agents that would abrogate myocardial fibrosis, which is a big problem, ischemic heart disease, and all that kind of stuff, as you know. And what I thought was unique about this is that not only did they, in their schema, include the ability to identify in an in vitro context, in sort of an in vitro disease modeling context, drugs that would abrogate that effect, but they also included a screen for toxicity. And the screen they used were iPSC-derived cardiomyocytes and endothelial cells. And in those cells, they measured viability, and they also looked at the force of contraction in the cardiomyocyte. So on the great side, they recognized that ultimately the success was going to be a balance between safety and efficacy, and so they included an ability to see a potential toxicity, particularly when they were looking for drugs that were going to be cardiac active, right? Cardioactive, because that's what the target was. So it makes perfect sense. Looking at cell viability, obviously makes perfect sense. But if you put in the context all of the ways that I just shared with you that the cardiovascular system fails, this is a very, very small subcomponent of it. And quite honestly, this is going to look for the real hammer toxicities. And honestly, those toxicities would probably show up in the initial screen of the myofibroblasts that they were using to do their disease modeling. So I'm saying all that to say, it's not unusual for folks to measure those kinds of endpoints in those kinds of cells as a cardiotoxicity screen. But recognizing our in vivo experience with the way that toxicity can play out in the cardiovascular system, this does not provide much confidence to the end user, particularly if that end user is a regulator whose job it is to protect patients or people, that in fact you're covering the full spectrum of the way that's going to happen. And I think that's interesting. Now, in this particular case, this context of use, pretty reasonable approach. But if you were going to use this to ultimately go to a regulator and convince them that you've got both an efficacious and a safe molecule, you wouldn't have much impact. On the flip side to that is that there are approaches that we commonly take in drug development that are representative of sort of a failure, a mechanistic failure mode approach. And this is known as secondary pharmacology screening, where we've identified specific sort of biological processes, receptors, endpoints, enzymes that we know are influential in cardiovascular function. And then if we screen for those kinds of bioactivities, that at least raises the potential specter that, hey, look, there's a possibility that you're going to have a cardiovascular effect, which generates a hypothesis that allows us to test that. So this is an example of a failure mode-based mechanistic screening approach, leveraging what we understand about cardiovascular biology. And in fact, we used it at NIEHS when I was in the Division of Translational Toxicology. So we took this approach and mined the ToxCast and the Tox21 databases and looked for specific assays. Now, so these are assays that were never designed specifically to screen for cardiovascular toxicity, but there are a number of assays in there that we know relate to bioactivities that might have a cardiovascular impact. And so we went specifically looking and to generate hypotheses that there were chemicals in those databases that had been tested in those assays that have a better chance than not of having a cardiovascular impact. And so now we're working through confirming those hypotheses that were generated. So this is us taking that failure mode mechanistic screening approach and applying it to environmental chemicals using data that currently exist in those high throughput screening databases. Another approach is sort of flipping it over and looking at the bioactivity of the xenobiotics themselves. And so this is a paper that is one of a growing series of papers that reflects on our experience in characterizing things that we recognize to be cardiotoxicants, cardiovascular toxicants in this particular case, and the modes of action. And so we've come up with a discrete list of things that are often associated with these particular drugs. And there's a paper for key characteristics of carcinogens and endocrine disruptors. And so you've all probably seen these papers. So again, this starts to give you some insights into the kinds of endpoints to be measured in the biological scope that I've shared with you previously that allows you to start building some confidence that if there is a cardiovascular risk, that you're likely to catch it because this is our experience in doing that. The beauty of the AOP frameworks, and obviously I didn't have anything to do with these particular frameworks, and I like them for a couple of reasons. One is that they represent sort of pathogenesis, and I like that because as a pathologist, that's the way I think, even though I don't often go back to the molecular initiating event. The other thing this does is build a bridge between some of those earlier key endpoints, key molecular events, and the ultimate apical outcome. And so if you could convince folks that, hey, look, by measuring those key events, I can predict that you're going to get that apical outcome, then I think you have a potential for bridging from those mechanistic outpoints to the apical endpoint without actually having to model it. The difficulty with this is that those relationships are oftentimes quantitative. And I think that's a scientific gap for us, is understanding that the quantitative relationship between how much L-type calcium channel blockage do I need to have before I'm actually going to see heart failure clinically. That's the rub, is the quantitative relationship between those two things. But it allows us to draw a line of sight between something that we know can be problematic in the cardiovascular system, in this particular example, because we understand the biology of the cardiovascular system and how it works, and ultimately the thing that I'm worried about. And it also is a framework for identifying where you actually build the assay system. And this is ongoing. So I'm not introducing anything new here. So the current paradigm is one that, again, it uses very complex biological modeling systems, animals. And we even compensate for the fact that we recognize that no one animal is going to be predictive of the human outcome and use more than one animal species. Those species generally represent relatively normal biology. That's not entirely true. There's background lesions that we know and recognize in those particular species. You put your xenobiotic on there, and then ultimately we look for what we recognize as how toxicity manifests. And then the outcomes of that inform the kinds of decisions that hazard assessors and risk assessors are trying to make. But it's problematic because of the throughput and lack of translation in some aspects, those kinds of things. I think that the frameworks that I've just shared with you are an opportunity to bring some structure into the way that we develop assay systems, the kinds of endpoints that we measure, and ultimately how you rationalize the bridge between those endpoints and the apical outcomes that we care about the most by considering what we understand about the biology of the target organs, which target organs are most problematic, the modes of actions of the chemicals or drugs that have produced the kinds of problems that we're interested in, the connection between key events and that apical outcome. All of those kinds of things are the kind of context we need to bring into our system, make sure we're modeling the appropriate breadth, measuring the right things to convince a stakeholder that, hey, look, these measures are ultimately predicting the outcome that we're concerned about. So the future paradigm is one that's going to include some high throughput screening, some biological modeling kinds of systems, measuring the appropriate endpoints, and all those kinds of things. Now, putting that in the context of the Precision Toxicology project, because it is the ASPIS cluster project that I know the most about. The reality of it is most of the things that I've just talked about are represented in that particular approach. The idea of having the biological complexity that is relevant to the human in vivo system. One of the things I like about this particular project is this notion of evolutionary conservation, right? So that helps you to recognize the bioactivities that are most likely to be meaningful because they're conserved across the species that are being evaluated there. The other part of that, though, particularly as we start to measure these more molecular endpoints, is understanding points of convergence of toxicity. I don't think we can reasonably design a system that is based on molecular initiating events. I think there's just too many ways a toxicant can be bioactive and ultimately lead to the kinds of things that we're worried about. But I do think there are sort of final common pathways of toxicity that are places that we can measure that ultimately give us confidence that, hey, look, this is going to go where I'm concerned about in a much more convincing way. The kinds of things that are being measured are relevant to the kinds of organ systems that we care about. The organ systems are some of those that actually float to the top when you start looking at the target organs that are most often affected. I think that part of what we need to do is to look at the kinds of endpoints and the organ systems and looking at it in the context of all the things that we think are potential targets in a whole mammalian system and make sure that we've got the appropriate breadth. We also need to understand how the kinds of pathways that are being measured are transitioning from an adaptive to a maladaptive stage. That's the point that goes from just bioactivity to adverse bioactivity. I think that's something that's going to take some learning and some experience in generating this kind of data and linking it to the kinds of apical outcomes that we're traditionally measuring now. So recognizing that, two things. One, the Precision Toxicology project has recognized that the context of use of even that particular approach is going to evolve. So it may have immediate usage in a screening kind of a paradigm because of the kind of information they can generate. But ultimately, it's going to generate hypotheses, and those hypotheses can be tested in more complex systems that ultimately is more informative from a hazard ID and characterization. So, and as we do those things, we'll learn more about the connections between those early events and the apical outcomes. And so at the NIEHS, we designed what we called the translational toxicology pipeline. So we had all of these capabilities and we would do them daily, but they would be done on different components. So not everything we did a literature-based systematic review on, did we actually do some in vitro screening or even the in vivo studies. So one of the things we intended to do was to deliberately work this paradigm so that every time I did some hypothesis generation through a bioactivity screen, I actually got the ability to understand its translation into that more complex in vivo system. And because epidemiology is such an important part of environmental hazard assessment and even ID, we have the ability to understand the sort of human outcomes of some of those things. So working the process increased our understanding that allowed us to be able to increasingly build confidence in those assay systems and understanding how to apply them. So boils down to this. I think there are some scientific gaps in all of this. I think we need to do a better job of defining the specific questions that we're trying to ask. We need to get agreement by the ultimate stakeholders that those are, in fact, the right questions. We've let ourselves off the hook because we got an animal system that has all the bits and parts in it, and we had never had to do that before. I think that's something that's missing in our current approach. I think to make that tractable, I think we need to leverage our experience in modeling toxicity and understanding which target organs are more often than not to those points of toxicological convergence that are going to be that sort of exposure or dose limiting target. Understanding the modes of action of the things that we've studied in the past. Get a better understanding of pathogenesis of some of these early events. The reality of it is that most of the in vitro kinds of things that we're doing aren't taking things out 9 months or a year or 2 years in the way we traditionally do animal studies. We need to get a better understanding of temporal pathogenesis and how that relates to those apical outcomes. You know, one of the great things about studying pharmaceuticals is that every pharmaceutical that's on the market today has the potential to be toxic just from a hyperpharmacology perspective. And so that's a huge database of experience and understanding how xenobiotics interact with a host that we care about that we can leverage. I'm not going to go through this in detail other than to say that, you know, I think we're doing a great job of building assay systems, measuring endpoints at levels of resolution that extend far beyond my ability to understand how to relate them to what's actually happening in the host and predict the ultimate outcome. But I think there's an incredible amount of novel insights to be gained from that if we ultimately give ourselves the opportunity to do it in a meaningful and holistic way, and then learn from the experience so that we progressively build confidence. And so, you know, 20 years from now, we're not still having this conversation, which is something that I fear based on our current approach. So with that, I'm going to stop sharing and give you all the opportunity to beat up on me.
I'm sure that's not going to happen. Thank you very much, Brian, for a well-delivered and thought-provoking presentation. We open this webinar now for questions. So just type in your questions in the chat or in the Q&A box, and I'm happy to read them out loud. The first question already reached us 2 days ago by someone who cannot attend this meeting today. So I'm happy to read out the question by Katja Lacasse, who works for Scientifique and is also part of the EPAA. I very much like the pillars of hazard approach, which identified bioavailability, bioactivity, adversity, and susceptibility as key for determining a hazard. And this question, just for everyone, is in regards to the paper that Brian Berridge introduced, the biological question-based approach. It's great also to see that such an approach covers the adaptability aspects, unfortunately often overlooked. However, I am missing the differentiation between some of these pillars applying to cell level versus organ or even organism level. This is particularly visible with, for example, homeostasis, an ability of the organism to adapt to a chemical exposure and not leading to adversity, thus at the level of the organ or full organism. Unsure is though if the adaptability effect would be visible at the level of the cell. It appears thus that there is a mix of toxicity levels that the approach may not cover. Any views how to approach this?
That is a great question, and actually one of the scientifically most vexing parts of this. It's the thing that keeps me coming back. The reason why I think we're so dependent on the description of the apical outcomes is because I don't have to know that. I don't need to understand the transition from adaptive to maladaptive because I'm describing the maladaptive response. And moving upstream to do this novel approach requires that we have a much better understanding of that, particularly when we're not going to replicate the sort of temporal components of the pathogenesis of most of the toxicities. I don't think it's going to be very tractable to run 28-day, 3-month, 6-month, 9-month studies in vitro. That said, I don't think we have to. Then part of my reason for bringing up this issue of final common pathways, and John and we talked about this yesterday, right? There's a molecular pathogenesis to be had here. That as you progress in injury, even at the cellular level, there's going to be the triggering or the initiation of pathways that are representing that sort of maladaptive response. One of the things, and I'm not a molecular toxicologist, and that's probably obvious, but one of the things that's always intrigued me is when I've looked at transcriptomic data from the heart, oftentimes we will focus on the things that are upregulated. And it's surprising to me how oftentimes, particularly in the context of overt toxicity, things are downregulated. I think we have to gain a better appreciation for what downregulation looks like. I think that's maladaptive. When things are supposed to be upregulating and responding, so when they got to a point where they just can't do that anymore, then you've obviously tripped into a maladaptive stage. So I don't have the answers to how to do that, but that's absolutely something that I think we need to intentionally educate ourselves better on to be able to get to this future state that's going to allow us to be more predictive. I think the way that you do that is to give yourself an opportunity to continue, even at the cellular level, of linking those molecular changes, transcriptomics, proteomics, metabolomics, whatever your omics choice is, to an apical outcome that is in vivo relevant. In the viability is not a good one because you've already pushed the cell too far. And that's the beauty of the cardiovascular system, is that there are sublethal functional things you can measure that allow you to have that sort of apical outcome that we recognize and are comfortable represent pathology, dysrhythmia, a decrease in contractility, those kinds of things. The other part of that too is, and we've tried to do a little bit of experimentation with this too, is actually stressing the system. So one way that maladaptive responses manifest is your inability to respond to a stress. We did some experiments with using dobutamine and shifting the dose response to dobutamine as a stress induced by a drug or a chemical, those kinds of things. So I think those require novel systems, maybe a stress system that allows you to see a subclinical change in that cell's ability to respond to that kind of stress. So that's a long-winded way of saying I don't know what the answer to that is, but that is an absolutely critical part of something we need to understand better to get our stakeholders comfortable with this is going to lead to that.
Thank you, Brian. John, do you also want to chime in on that question? I'm sure you have thoughts on how to use omics data.
In fact, what I very much enjoy about working with omics data, it's a comprehensiveness in terms of what you get as a result. You ended up pointing out, indeed, Brian, something that is falsely stated in that document, that scientifically there are no barriers. In fact, I think that the mistake there is I had synonymized scientific with technological.
Yeah.
Where technologically speaking, we're in a situation where this kind of approach and this kind of revolution can take place. And I like to quote something, Sidney Brenner, to my students, who in 1980 ended up saying that progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. What he was really referring to here is that up until the omics revolution, we were very much hypothesis driven. We would ask the question, does this chemical cause that adversity? We're in a situation now where you can actually collect information about the biological system, where you can begin to ask the question, what adversity does this chemical cause? Or even better yet, what chemicals if you want to go talk about throughput now, cause these adversities.
Yeah.
So you end up actually becoming a little bit more different in the way of thinking. So really where the challenge is that other component that you were referring to, you ended up actually asking the question, well, how many target tissues do we need to use? And in essence, that's the biology, right? So we can capture a lot of the functionality of that biology, but good luck trying to pull out a neurotoxin if you're using your Hep G2 cell lines in order to get at that answer. So then you have to kind of like merge the ideas where what do you need to represent biology as a test system?
Yeah.
And what kind of technological approach do you take in order to measure that, as you pointed out, the signal of the pathogenicity? I think that's where really the challenge lies, and it's the same thing as well with respect to that transition, Leonie. Depending upon when you test, will give you different outcomes as well. And that transition that you're talking about is a very, very early stages before you go from a compensatory response to one that deviates completely away from anything that the biology can deal with. And so how do you capture that point of departure where you're going into, that failed state as opposed to we'll normalize this as much as possible based on the options that I have. And so I think that conceptually speaking, what you have provided in this framework, by asking the questions, you're kind of like reducing that complexity into things that must be known about the system in order to say something about toxicity. And I like that very much. And I think the greatest challenge that we're currently facing with the PrecisionTox really is that we're using omics data, we're using metabolomics data, so we have that richness of the data, but we're getting profiles. We're getting things that basically show us the biology, but I'm told by my regulatory colleagues that unless, of course, you can describe that molecular biology as in a toxicologically interpretive way, then it's really difficult to make regulatory decisions around it as well. So in that biology, you will see that compensatory stuff. You do not want to basically regulate based on the compensatory aspects of the signal that you're getting. You want to regulate based on a signature of we're on a path towards some adversity. How do you identify those and how do you end up actually using those in a regulatory context? I think that's a big challenge that hopefully in the near future we'll be able to solve.
Yeah, and I again, that's my optimistic view of the world, but I think that if we stopped long enough to think through these different organoids, and again, I use the cardiovascular system as an example. We know a lot about the cardiovascular system, and we know the fundamental, most important sort of biological processes that make the cardiovascular system. I think we have the ability to articulate those things. We just haven't done that. And so you just have to keep replicating the apical outcome. And I think that's where we're kind of stuck. The other part of that too, I'm sorry to interrupt you, John, but the other part of the questions thing is again, recognizing that everybody has got a different idea of the questions in their head. That articulating the questions allows you to agree on that's the question I really need to answer.
Good. I am monitoring the chat and QA. So far, nothing has come in. So I take the chance to ask a question to you. And it is about one of my favorite topics to talk about, specifically in the regulatory context. And that's how to advance the AOP framework, how we do it, what experience we do, and what data we gather. So it can potentially be more relevant for regulatory science. I am very partial, just a disclaimer, to this AOP framework, because when I first started going to CTAC conferences during my studies, this was the new thing that was introduced. It comes from ecotoxicology. It made very much sense to me back then to setting up such a framework to collect the data that we generate using NAMs for them to potentially one day become relevant for regulatory aspects. So you touched upon what you think should change in regards to the AOP framework already in your presentation, that is adding this quantitative aspect to it. Is there more that you think that needs to be added to this framework? And also, does the AOP community know that they need to add like specific things? Because there is so much time and money and effort that's going into building these pathways. And the AOP wiki is filling up like day by day. And I would really, like, I'm a bit heartbroken sometimes almost when I see that the love for AOPs is not shared by everyone. So that's my questions. Like, what can we do to advance this?
It's a good question, Leonie, and I have confessed to you that I like it conceptually. I like AOPs conceptually because it represents pathogenesis, and that's just the way I think. That said, I think the current approach that is largely focused on molecular initiating events is an exercise that you'll never finish because I think it's too broad. If I had to develop an AOP framework, I'd start with the failure modes and I'd come back and I'd trace them back as far as I thought I needed to get to a key. John likes to talk about nodes in the context of gene expression, a node that represents that transition to a maladaptive response. And that's where I would focus my building my systems and measuring my endpoints. Now, it just so happens the example I use is a great example because it sort of does that. We know that the L-type calcium channel is absolutely critical as it relates to contractility in the heart. So that's a good one. That's why I use it so much. But there are lots of other AOPs that are things that I don't know what to do with because they're just too complex or they go back too far. So that's a long-winded way of saying that if somebody handed me, and I've had to do this with some of my clients and who are developing systems, is to try to start with, okay, look, you're developing a liver system. This is the way the liver fails. Okay, and so what is it? What leads to those kinds of failures? Be it biliary necrosis or hepatocyte necrosis or phospholipidosis or any other ways that we know that the liver can be injured, free radical generation, those kinds of things. So start with those things and then walk back the pathogenesis to a place where you feel like that you can model it in an effective, efficient way that's not so far back that I can't make the connection between those two because there's too much math between the molecular initiating event and the likelihood that that's going to be an apical outcome. So I'm with you. I like AOPs, but I would start at the apical end and come back rather than the molecular initiating event and go forward if I had to do it.
Thanks, Brian. John, do you want to add to that, or in general, just open up the floor now for you and Brian to have an interaction about how the different approaches can advance chemical safety assessment using NAMs? Because I have looked at the chat and the Q&A, and apparently everything you say is so crystal clear. There are no questions or comments coming in. So,
and we all know that's not true. So
well done, Brian. I completely agree.
So I'll just leave it up to you now to engage with each other's positions, and I'll only interfere if there's questions coming in from the audience.
It's nice to see that there is a question from Susanna.
Isn't that great? Okay, I will read it out loud to everyone. Thank you so much for the presentation. It highlights really important challenges that have been blocking further uptake of NAMs. Thresholds of toxicity or internal threshold of toxicity seem to be a framework that would be relatively useful and easy to implement in chemical risk assessment. Do you see value for it, specifically for cardiotoxicity, but also in general? Thank you, Susanna.
Yeah, no, I think that the principle, I use cardiovascular because, again, that's the system I know most about, but I think those principles apply across the target organ systems of interest. You could use the key characteristics papers as a good sort of foundation because that's essentially what they've been doing is kind of thinking. Now, they've been thinking about it from a mode of action perspective, not necessarily the failure mode perspective, but there's a line between those two. And that ultimately is something that can be leveraged. So And the short answer to the question is, I think that those principles absolutely apply across the target organs. The key challenge, another key challenge, is deciding which target organs. I mean, I'm looking histologically at 42, 43 different tissues. Are we really going to develop 42, 43 different modeling systems? I don't think so, but I don't think we have to. And that's where I think if you went and looked at our, sort of reflected on our experience, you just find out that there's probably half a dozen organs that are going to catch most of the problems that we're dealing with. So I think it can be applied beyond the cardiovascular system. Great question. Thank you for that.
And of course, you're reading the question very much as a European, as an American, where risk is part of the framework, but in of course, Europe, it's mostly hazard-based. And the challenges, I think, are equal in both cases, right?
Yeah.
Because if we go back to, for instance, the measurement of bioactivity, one of your 4 pillars.
Yeah.
In both cases, you're going to be utilizing readouts that are informative of mechanism, and let's just say it's going to be omics kind of readout. And if you, instead of basically, if in this case you're basically measuring the omics responses in a dose-responsive manner, and you're going to ask the question, at what point do you start seeing a response that is indicative of adversity, and set your thresholds there in terms of your internal or external thresholds of toxicity. You're still going to have to decide, where do you fit that threshold? Should it be the first gene that shows up as being dysregulated? It doesn't make very much biological or toxicological sense to do so. If you, in the literature, you'll find people who make somewhat arbitrary decisions will take to, the 50th gene that shows up. And I think that as we get more and more rapprochement between the toxicological questions that we're asking and the biological or biomolecular information that we have tied to a disease state, we can basically be looking for, as you pointed out, your within your failure modes of action, at what point do you basically see something that's identifiable as a failure?
Yeah.
And so I think that in one case, you can imagine that if you're going to start asking the question, where do you draw the line in terms of toxicity threshold, you may very well decide to draw the line as soon as the gene set that you've captured is really recognizable as a biological component that has gone awry. Endocrine disruption would be a great one. I mean, endocrine disruption is actually a great example because the molecular phenotype is in fact the adversity that is being characterized as an apical endpoint. And so here in this case, if you actually have biomolecular markers of endocrine disruption, I suppose you could kind of like set the threshold as soon as the gene set that lights up as a rank order of sensitivity responses could very well capture that link between bioactivity and long last, not instead of having a bioactivity profile, but actually having a bioactivity signature by which you tie it directly to a toxicological outcome, even if it's also in an AOP framework.
Yeah.
So it's quite encouraging to think that your framework that you're describing, applies to both the hazard-based decision-making process that occurs as well as the risk-based.
Yeah. One part of that, and you and I talked about this yesterday too, and that is getting a better understanding of just normal dynamic range, right? Because all biological processes have a dynamic range. And so how do I know? I mean, the whole basis for clinical pathology, for example, is ALT, AST is present in everybody's plasma. But I know that a certain level of ALT in the plasma actually indicates something bad because I understand the dynamic range of that particular. I don't know that we've ever gone back and looked critically at the normal dynamic, natural dynamic range of a lot of these sort of molecular endpoints that we're interested in to know what exceeds that normal threshold, to your point. So the threshold could be either you're outside the normal range, whatever that dynamic range is, or it's gotten to a place where it's tripped some other process that actually is where the injury is going to happen, right?
It's fantastic that you mentioned that because in PrecisionTox, we're still accumulating the omics data.
Yeah.
And the way that it's constructed is that for every treatment, you need a control.
Yeah.
And we block it off so that you have as many treatments as possible tied to a control. But at the end of the project, we're going to have way more control samples than we have any particular of a treatment sample in terms of biological replicates. We may have sufficient to come up with, the normal ranges for each of these species in terms of what normality looks like. And once you do that, you can significantly reduce the number of animals to be tested because you don't necessarily always have to have that controlled background. You know what the background looks like. All you really need to do then is to basically measure your treatment and where it falls within the distribution of normality. So it's a Z-score statistical test as opposed to a
well, in fact, that's the way medicine's practiced, right? So I don't bring a control when I go to the doctor and they measure my blood chemistries. They know what the population range is, and I'm measured against that range. Now, that said, sensitivity comes in and knowing what Brian's usual range is. And when I get outside of my range, that may not be outside of the population range or different than the population range.
We have one more question from the audience. I'm just going to read it out loud because I'm unsure who can see the text and who can't. So this question is by Jay Dawick. I'm sorry for butchering potentially your last name. The bioavailability and bioactivity pillars of the biological question-based approach can be comprehensively addressed for most substances using non-animal methods, but addressing the adversity pillar for complex endpoints like STOT and DART is much more difficult. Read across two structurally similar compounds with available in vivo animal data will play a key role here, as it may be a less complex approach compared to, say, AOPs. However, demonstrating read across can also be challenging, as evidenced by the high bar regulatory agencies like ECHA set for acceptance. Thoughts from either Brian or John on this, and what tools like omics could be used to bolster and improve read across approaches to address questions on adversity?
I will say, one, that's another really great question. And from a pharmaceutical perspective, what undermines confidence in read across is recognizing that, for example, if you've worked for more than one pharmaceutical company, one of the things you recognize, particularly big pharma, everybody's chasing the same targets. Well, not everybody's successful, right? So somebody gets the molecule right, even though there's lots of molecules that have very similar designs. It doesn't take much of a tweak in that molecule to actually have liabilities associated with it. So read across has got some challenges of making inferences just because it looks similar to this, that the hazard profile is potentially, because it can be very small change in the molecular structure and have significant differences in the ultimate hazard profile. That said, I think in a general context, it probably can be more useful. But this is another one of those places where I think it's going to be an evolution. I think that read across is a great opportunity to generate a hypothesis that ultimately you can confirm or deny based on some experimental evidence that supports that hypothesis, as opposed to, I think it looks similar to this, it's going to be similar to that, and we should just buy into that. I think you've got to qualify that inference, at least until we learn enough to understand enough about structure activity relationships to be able to do that without that experimental evidence.
I love Jay's question. And it's probably the lowest hanging fruit, I suppose, that we have in using mechanistic omics-based data. One thing that I keep reminding myself about read across is that it first starts with grouping, as you pointed out, Brian. And typically the grouping is done based on structure similarities among the chemicals. But at the end of the day, it is but a hypothesis. And as a scientist, you want to basically either give evidence in support by trying to disprove that hypothesis. So how can you best disprove that hypothesis? Well, layer down more information. And how about laying down some biology onto it? So then if you had a read-across hypothesis that was solely based on structure, why not use omics data in order to then see whether the as your pillar says, the bioactivity profile is shared among the chemicals that form a group or not. So you're basically challenging the structure-based grouping based on biology data. And then the next challenge, of course, is going from that pillar to your next pillar, which is adversity. So we're back into this situation where you may be able to substantiate and show great evidence, both statistical and biological, around a group, allowing the read across to happen. But if you're solely going to base your regulatory decision around the bioactivity in terms of identifying what the hazard is, then all of a sudden we're back into the same challenge of interpreting it in a toxicological viewpoint. So I think that this is a grand opportunity to really see where, as you say, we can graduate the utility of these frameworks and graduate the utility of these technologies and our knowledge of AOPs by simply starting this process of at least giving greater confidence in the current approaches that we take, and then see to what extent we can begin to then start replacing it. But one thing that I'm quite intrigued by too is that whenever you have a grouping, if you choose the right chemicals to group with your target chemicals, so if you end up actually including chemicals, for instance, that you know what the biomolecular signature is of a particular toxicological endpoint, then that adds reinforcement as to if that chemical groups with your target chemical, you now basically have a comparative basis in terms of interpreting your biomolecular readout, knowing what the one that is actually known to be adverse because of this particular pathway, whether you actually see that grouping occurring because of those elements within your measurements, as opposed to some other random association of gene profiles.
Thanks, John. Now we do not have any more open questions, so you can just go ahead and have a chat for the last 10 minutes of this webinar.
Well, we still have quite a few people that are
Well, let me throw this out to you, John. One of the things that I've often thought about too, the current approach is very, very standardized, which allows us to make get a sort of collective experience, right? The way I do a 28-day study here in the United States is very similar to the way you do it. I mean, because we got to work a lot of international organizations. The NAMS world is not very standardized. We got lots of different assay systems, people measuring. There's probably more commonality in the things that are being measured than maybe the biological platform in which they're. At some point in time, I think that we've got to settle on a common platform and start generating common data that allows us to get enough collective experience to be able to rationalize how we get to that next place, building that sort of confidence, to Laura's social construct there. And I don't know what that, and to some degree, I think that is another reason to have a better sense of the specific questions, because then you can start to identify a platform, get everybody to agree to that's the one because it's got the right biology, I can measure the right endpoints, and I can draw a line from here to the thing that I care about. But as long as we're in this sort of discovery phase of NAMS, which we seem to have been in for over 10 years now easily, I don't know how you get to a place where you can get that collective learning enough to build that confidence, if that makes any sense. I think yeah,
it makes a lot of sense, because part of the PrecisionTox project is really to identify those barriers and to find solutions for them.
Yeah.
And part of it is, in fact, building that confidence, but it's also building that confidence in a framework that everybody already believes in or is engaged in. The OECD, for instance, is already starting to move in this direction through the work that we're doing and the work that one of our partners is doing, Michelle Health Science. Writing guidance on how to collect and report omics data so that it's more regulatorily relevant, for instance. How to do the same for sampling if you're basically going to be using a NAM technique. And I think it begins by simply understanding the process that is currently used to build that confidence without falling into the trap of demanding that that's the only process that we can build confidence or to validate or to normalize these approaches. Because one thing that we don't want to start doing is, for instance, setting the animal tests as the benchmarks by which we determine whether a NAM is useful or not. I believe that if we do this correctly, the types of data that we're obtaining is richer, right? It's more revealing, and so it tells us more about the biology than to the point where we can actually start making decisions around the biology, as opposed to making decisions around a particular observation, such as an apical endpoint.
You bring up a really good point, and then we've got another question, I think, here. And that is, and it's a common focus of debate, and that is using the animal study experience as our benchmark. And I think there's a difference in using that experience as a source of understanding about how toxicity plays out and the relationship between apical outcomes and particular upstream molecular events than using the animal studies as a benchmark. And I think the former has far more value than the latter, because you don't want to be constrained by a system that you think is actually less effective than the one you're trying to build. But that said, that is the basis for our understanding of what toxicity looks like and how it gets there. And so we need to use that experience.
We have one comment by Jay, who thanks both of you for answering his question, and says that biological-based read across using NAMs instead of just structural similarity is fully aligned with his thoughts, and he takes ownership of the acronym BBRA, which can be added to the BQBA.
There you
go. But then we have a question by Susanna on the topic of standardization of NAMs. Who can enforce that standardization of NAMs? There are some OECD guidelines, but even those are not fully followed.
That's a really good point, and I'll use as an example. So in the world of pathology, I mean, as you all probably recognize, pathologists are terrible about using different words to describe the same thing, and it was kind of a mess in drug safety assessment. And ultimately, the FDA said, "Look, you guys got to get your act together, because I'm seeing lots of different words that we think are related to the same thing. We don't know because you all are using different words." And so there was an international effort called the International Harmonization of Nomenclature or something. I chaired the cardiovascular committee. And we basically had international working groups that all got together and said, "Look, if you got this lesion, this is what we're going to call it. And if you got this lesion, this is what we're going to call it." And so now we have a significantly greater standardization. Now it's a flexible system, because new terms come up and people recognize that there are things that we need to do differently. As you introduce different kinds of therapeutic modalities, they produce novel manifestations of toxicity and stuff. So it's got the flexibility to it. But the community got together and decided that this is the way we're going to do that, and that's had a huge impact on the regulators' ability to understand what we're trying to tell them. So I'm saying that to say that one way to do that is to expect a regulator to tell you exactly what to do. They're probably not going to do that. The FDA certainly won't do that. Or the community can step up and say, "Look, it would be to all of our benefits if we all collectively decided that this is the way we're going to do this."
I agree with you, Brian. And this is not anything new here, because of course everything is being standardized right now, primarily driven by trade. There's a reason why we can take our plugs from one country and expect to, buy a converter for our plugs and expect to see that on the other side. It's all standardized. And I do that because I've been having some problems charging my computer because of a failed power pack. But Having said that, Brian, I think that that's a structural thing, right? We have to actually fund the structures to make that happen. And I know that in Europe and in North America, there are these reference laboratories where you end up trusting an expert group to validate it. There's not enough resources that are going into these structures. And if we really wanted to, if we were serious about making this happen, I think that we have to look very seriously at funding things that are typically not funded. As we're reminded, the European Commission has spent €1 billion over the past 10 years or so on the science of new approach methodologies, but how much of it has actually gone into taking that knowledge and making it applicable? That would be an interesting number to have, and I'm sure there's going to be a small fraction of the discovery process monies that have gone into it.
Well, certainly we've stimulated an economy around the development of NAMs, but have we really impacted the way that we do chemical hazard assessment or drug safety assessment? I think that's a much smaller impact.
Or have we really appreciated what the economic consequences could be? And I keep again saying this to my students. Soon after the Human Genome Sequencing Project, and we're talking about within 10 years, there's a $50 billion industry on biomarkers that was derived from that knowledge, right? And wouldn't it be wonderful to actually have a European €50 billion industry built around chemical safety biomarkers?
Yeah.
There's an incentive to do this in a sense, because with that comes intellectual property, where then you can see a whole bunch of SMEs coming out of the grassworks and basically creating this momentum where all of a sudden it's not pushed from the public side of the equation, but it's being pulled by the private side of the equation in terms of new innovations, but also the translation of that new innovation should actually be privately funded in the end, because there's an incentive to do it.
Yeah. If we've all agreed on the sort of standards that need to be in those systems and the things that we're going to measure and all those,
And of course, that's
in fact, we in the microphysiological systems working group, we tried to do that. We published a series of papers that were looking at specific target organs and outlined the biology that needed to be represented in those, and the endpoints that would be measured, and how those endpoints would be used, and those kinds of things. I think we need more of that kind of discussion.
Mm-hmm.
We have one final question. If I could ask for just a short reply from both of you, in case you are aware of what Karen Tillmann is referencing. Hi, John and Brian. Can you see value in using the ETAP? And this is the EPA transcriptomic assessment product approach for pharmaceuticals.
My answer will be short, because I was not aware of that, and I'm going to learn about it, so I don't know.
My answer is absolutely yes. And here's what's very interesting. This is aggressive. This is like, let's collect what the transcriptomes look like when exposed to these chemicals. And I think that the EPA in North America is a little bit different from Europe in that regard, because I think that there's a lot more willingness to actually make decisions and around bioactivity profiles, where if you were to group based on this kind of transcriptomics data, it has weight, as opposed to absolutely necessarily tying it to adversity. And so I think that there's a more, would I say, more liberal view of what the transcriptomic data represent. It could be indicative, let's say, of toxicity, but maybe in Europe we need it to be a little bit more predictive. And so there's a dynamic there that I'd like to see evolve, and to see whether we can come up to a consensus as to, because it's high throughput, how much can we actually say about toxicity simply by having amassed a huge amount of information on the biomolecular responses to the exposure to these compounds.
Thank you, John. And I think with this, to stay within the time limitation that we have for this webinar today, we are concluding this webinar. Thank you again, Brian, for your presentation and engaging with the audience questions, and also with John's PrecisionTox perspective. John, thank you for participating today as well. As always, it's a pleasure to hear your insights. And thank you everyone who joined us today. We will be sharing the recording of this webinar after on. If you missed part of it, or if you think it's so interesting, you want to send it to your colleagues. And Thank you to everyone involved today and take care. See you next time.
Thank you, Leonie.
Thank you,
Bye,
Leonie.
everybody. Bye, John.
Good. John, yeah, I will save the recording. Again, thank you so much for your insights today. There's
How did
still
that go?
some attendees in the room. Hope you have a good rest of the day.
Thank you, Leonie. Bye-bye now.
Talk to you soon.